Joseph Priestley discovered that plants restore to the air whatever breathing animals and burning candles remove. John Ingenhousz showed that sunlight is essential to the plant process that somehow purifies the air fouled by burning candles or breathing animals. Julius von Sachs proved that the green parts in plants is where glucose is made and that the glucose is stored as starch. T. W. Engelmann described first action spectrum of photosynthesis and also observed that the bacteria accumulated in the region of blue and red light of split spectrum. Cornelius von Neil demonstrated that photosynthesis is essentially a light-dependent reaction in which hydrogen from a suitable oxidizable compound reduces carbon dioxide to carbohydrates. Photosynthesis requires a lot of pigments which trap solar energy. Major pigments involved in photosynthesis are chlorophyll A, chlorophyll B, xanthophylls, and carotenoids. Light reaction involves different steps. First step is absorption of light by pigments. Pigments present in chloroplast trap solar energy, and photosystem 2 present in membranous part of thylakoids is involved in splitting of water molecule. As a result of splitting of water molecules, hydrogen ions and oxygen and electrons are formed. So oxygen is released as a byproduct. And as a result of electron transport chain, high energy chemical intermediates like NADPH and ATP molecules are formed. Electron transport chain involves two major photosystems and a series of intermediates which are involved in electron transportation. First photosystem 2 absorbs light, it becomes excited and its electrons jump to higher levels and then are transferred through a series of intermediates to photosystem 1. Cytochrome B6F is the major intermediate. Then photosystem 1 becomes excited and it transfers its electrons to ferredoxin. Then from ferredoxin, hydrogen ions reduce NADP to NADPH and some of the electrons are transferred back to cytochrome B6F. Since this is cyclic flow of electrons, because electrons are transferred from ferredoxin to cytochrome B6F, then to photosystem 1, and then same flow occurs. This is known as cyclic photophosphorylation, and ATP synthesis takes place. Chemiosmosis involves maintenance of proton gradient. In case of photosynthesis, proton gradient is maintained in such a way that hydrogen ion concentration remains higher towards inner side of thylakoid and lower towards outer side of thylakoid, that is in stroma side. This is because splitting of water molecule occurs inside lumen of thylakoid. Concentration of hydrogen ions remains higher and then electrons are transported by electron carriers, and during each electron transfer protons are transported from stroma to lumen of thylakoid. And one more reason of this proton gradient is NADP is reduced to NADPH towards stroma side, and this results in accumulation of protons in lumen. Now breakdown of this proton gradient is by ATP synthase enzyme, which has two units, F0 and F1. F0 unit acts as a channel of protons and F1 unit is the catalytic part which is involved in synthesis of ATP from ADP. So phosphorylation takes place. Calvin cycle involves three major steps: carboxylation, reduction and regeneration. RUBP fixes one molecule of CO2 in the presence of Rubisco enzyme. and a 6 carbon compound is formed, which is highly unstable and breaks down to form 3 phosphoglyceric acid. This phosphoglyceric acid is reduced to form 3 phosphoglyceraldehyde, and this step is called reduction. And after this, RuBP is regenerated and simultaneously glucose is synthesized. For synthesis of one molecule of glucose, 6 CO2 molecules need to be fixed. C4 cycle is known as C4 cycle because the first formed product of C4 cycle is a 4 carbon compound called oxaloacetic acid. In this case, plants have two types of cells, mesophyll cells and bundle sheath cells. In mesophyll cells, phosphoenol pyruvate fixes a molecule of CO2 and forms a 4 carbon compound called oxaloacetic acid. This oxaloacetic acid is isomerized to form malate or aspartate. Malate or aspartate. These are then transported to bundle sheath cells where these release CO2 and form a 3 carbon compound called pyruvate, which is transported back to mesophyll cells. So PEP is regenerated, which again fixes carbon dioxide in mesophyll cells, and the CO2 which is released in bundle sheath cells is then used for fixation of carbon dioxide or for C3 cycle. So in case of C4 cycle, both cycles operate. In this case, two cells are there, and C3 cycle occurs only in bundle sheath cells. There are many factors affecting photosynthesis. These can be divided into categories: external and internal. External factors include availability of sunlight, temperature, CO2 concentration, and availability of water. And internal factors include number, size, age, and orientation of leaf, mesophyll cells and chloroplasts, internal CO2 concentration, and amount of chlorophyll. If we discuss these in details, availability of sunlight, the availability of sunlight if increases, generally intensity of light increases, then rate of photosynthesis increases. And we can say quality and quantity of sunlight both affect the rate of photosynthesis. And now in case of temperature, it has been observed that when temperature increases, rate of photosynthesis increases. But this increase is observed only up to optimum temperature. Beyond this optimum temperature, rate of photosynthesis decreases. Then CO2 concentration. It is observed that when CO2 concentration increases, rate of photosynthesis increases, but this is up to a certain limit. Beyond that, it becomes toxic. And availability of water, when water availability decreases, rate of photosynthesis decreases. And internal factors, number, size, age, and orientation of leaves. If number of leaves is more, since leaf is the kitchen of the plant, uh, rate of photosynthesis is more. And mesophyll cells and chloroplasts, in case of C4 plants, we observe two types of cells, mesophyll and bundle sheath cells. So rate of photosynthesis is more. So anatomy of plants is also important in determining rate of photosynthesis. And internal CO2 concentration effects. Internal CO2 concentration means plants utilize CO2 which is released as a result of respiration process, and an amount of chlorophyll. It's obvious that if amount of chlorophyll is more in a cell, rate of photosynthesis is more.